Yeah, very good morning to one and all. The next topic is nonlinear pharmacokinetics. This topic is under Unit 6 of your syllabus copy. So, I completed till Unit 3. After Unit 4 and 5, you have many derivation part. You have many derivations part. So, that's why after that I will start this 4 and 5. So let I will discuss it about unit 6 non-linear pharmacokinetics. So what do you mean by non-linear or linear? See ma'am, once we take a drug, first we take a drug in the form of dosage form. Then it will undergo absorption, distribution, metabolism and excretion phenomena in our body. So in between this drug will exhibiting all these parameters, it will be dependent upon its dose, right? It is not depending upon the time. These are not time depending processes. Only these processes will be occurred in our body after taking a dosage form which is consisting of a drug. It will be dependent upon the dose. Then it will be exhibiting some ADME mechanisms in our body. Meanwhile, it is traveling in our circulatory system dependent upon the volume of distribution and Ke elimination and AUC, like these parameters. And this type of mechanisms which are the drugs will follow, this type of mechanisms, these drugs may undergo linear kinetics, or those drugs may follow some linear kinetics. So for such drugs, when you take the plasma drug concentration levels of the drug and the time, we will get a linear graph like this. Either it may follows zero order or first order. Okay, so if it will be zero order, it is concentration independent. If it is first order, it is traveling around this concentration dependent. So we can tell depending upon the concentration of the particular drug, either it may be dependent or independent, but it will follow some linear kinetics when you take the concentration of the plasma drug concentration of the drug and the time. Okay, so it will be dependent upon these types of pharmacokinetic parameters, and these will imposing all these mechanisms of the drug in our body. Then they will follow linear kinetics. Then what do you mean by non-linear kinetics? The name itself indicating that these are not following linearity. Why these are not following linearity? I already told you, once you take the particular dose of the drug, these may be traveled around these mechanisms. That means there may be a chance of this irregularity of these mechanisms of the drug. So because of the irregularity or because of the saturation of any one of the phenomenon among these four mechanisms, then the drug can able to follow this non-linear kinetics. See, if the drug will follows it dose dependent, or if the drug will follows this ADME mechanisms as you described under pharmacokinetic parameters, either it may be given by IV bolus or intravenous or oral route, whatever it is, but the drug will follows this mechanism, it will get a linear graph when you take the concentration and time relationship. But in case of non-linearity, there may be a chance of any one of the phenomenon of this ADME will get be saturable, or there is some fluctuations in distribution or metabolism pathways. In that manner, few drugs will undergo such kind of kinetics. Those are called non-linear pharmacokinetics, and these are also called as capacity limited kinetics. Capacity limited, that means this type of drugs will having some limited capacity, either there may have some saturation with absorption or distribution. If the drugs will have saturation with protein binding or the distribution, definitely they may have some capacity. If they are available in such, suppose 10 mg of the dose, this will be distributed in 100 ml of body fluid. Suppose you just assume it. So in case of this capacity limited, here 100 ml of this fluids are not available, only 70 or 80 ml of the fluids will be available, definitely it will be saying up capacity limited kinetics. So that's why this nonlinear kinetics are also called as capacity limited kinetics. And one more important name for this nonlinear pharmacokinetics is mixed order kinetics. These are very very important, ma. In your exam point of view, either they may be asked you like mixed order kinetics or nonlinear or capacity limited. These three are synonyms to each other. So these are also called as mixed order kinetics. Why these are also called as mixed order kinetics? See, in some cases it will follow zero order, and in some cases it will follow first order. How and why it will follows? Suppose your drug is showing the T half like 7 hours. Suppose your drug is showing the T half of 7 hours. Okay, the elimination will follows for this drug is by first order, and before that absorption and distribution phenomenons will also occurs through zero order. So up to 3 to 4 hours it will showing some zero order kinetics because of the particular drug absorption and distribution. And in case of elimination, it is eliminating out through the first order kinetics. This thing was practically I observed in case of phenytoin sodium when I was done with my MPharmacy project work. Okay, while doing that MPharm project work, I made a phenytoin sodium sustained release tablet, ma. Okay, phenytoin sodium sustained release tablet. So I observed this kind of kinetics in phenytoin sodium. Actually, this is having the half-life of more than 7 hours. So I used to get every time mixed order kinetics. That means the drug is not properly released in vitro dissolution studies also. So when I plot a graph between the concentration data versus the time data, I used to get two curves, zero order and first order, like this is called zero order and this is the first order. Sometimes it is showing this straight line curve, this is of first order, right? And this is of curved line, it is showing zero order. So every time I used to get the curves like this. First I did not understand what is going to happen. Actually we know only this type of curve, then why I used to get two different curves, I did not understand. Then once I will check this phenytoin sodium properties through Google, I got to know something. Okay, it is following some non-linear kinetics. This is also called as capacity limited and it is also called as mixed order kinetics. So for that reason, this phenytoin sodium is showing these two graphs. See, ma'am, while doing the study of in vitro dissolution studies in case of tablets, while doing in vitro dissolution studies, definitely you can get these two curves. Either there may be showing some saturation with absorption phenomena or distribution phenomena or metabolism or excretion. So these are the main four causes of the non-linearity. Why the drugs will exhibiting this non-linearity? Because of these four causes. So for that reason, you need to learn the factors causing the non-linearity. They clearly mentioned in your syllabus copy. Factors causing non-linearity. So how we will derive these non-linear factors? With the use of Michaelis-Menten equation. He is a scientist. He developed the equations regarding this nonlinear pharmacokinetics. So whatever the cause will happening for this nonlinear, so we need to calculate those pharmacokinetics with the help of this Michaelis-Menten equation. So in your exam point of view, these two are very very important, and in the nonlinear pharmacokinetics, only two topics are there. First one is factors causing nonlinearity and the second one is Michaelis-Menten method for estimating the parameters. So the drugs, those are following this nonlinear pharmacokinetics, Though for those drugs, we can derive the pharmacokinetics with the help of Michaelis-Menten method. In this class, I am going to tell you about the nonlinear pharmacokinetic introduction and the causes of nonlinearity. See, first the relationship of linear pharmacokinetics. At the therapeutic doses, the change in the amount of the drug in the body or the change in the plasma concentration, which is due to the absorption, distribution or binding or metabolism or excretion. It is proportional to its dose. So these all should be proportional to the dose of the drug. Either there may have some amount of the drug in the body or there may be a change in its the plasma level, plasma concentration level. So when you administer the drug, either may be a single dose or maybe a multiple doses. It is not considered either it may be a single dose or multiple dose, but the relationship of the dose is directly proportional to the mechanisms of the drug. Particularly when it enters into the body, it will be exhibiting these four mechanisms. Okay. So in such situation, the rate process are said to follow first order kinetics or linear kinetics. When you take these pharmacokinetic values or concentration versus T, C versus T difference at different doses when collected for dose administered are superimposable. In case of this linear pharmacokinetics, this word is very, very important. Superimposable. So when you plot the plasma concentration of the drug under y-axis and this is of time under x-axis. When you take these two parameters in these axes, so what you are going to find out? We are going to find out C max, AUC, T max, okay, Ka and Ke, all these pharmacokinetics we can able to find out. When we can able to find out all these pharmacokinetic parameters, your drug dose will be directly proportional to the ADME mechanisms. Then we are going to find out those drugs pharmacokinetic parameters with the help of C versus T curves. C versus T curves, what I told you under second unit, introduction to pharmacokinetics. So these are said to be linear pharmacokinetics and these are superimposable. This superimposable is your ADME mechanisms are reflecting to the objectives, reflecting to the objects like Cmax, Tmax, AUC, all these etc. All these pharmacokinetic parameters. These mechanisms are going to reflecting all these pharmacokinetic mechanisms. That's why this is called superimposable. See, what do you mean by superimposable? If you take one object, it just reflect to other object, but these two will be visible. So same in the same pattern you have to think about this linear kinetics. These are superimposable. When you take consideration into ADME mechanisms, these will be reflect their pharmacokinetic properties in the form of either of Cmax, Tmax, AUC, okay, and then T half, biological T half, and so on. So we know up to this mark, okay, what we studied in the previous classes, we know up to this mark. And the next part is somewhat different, that is non-linear. No, see, the import, this is of linear only, this is of linear only. The important pharmacokinetic parameters, that is fraction of bioavailability. Capital F is fraction of bioavailability. Ka is the absorption rate constant. Vd, volume of distribution. Clr, clearance of renal. Clh, hepatic clearance. Fraction of bioavailability, absorption rate constant, volume of distribution, renal clearance and hepatic clearance. These all the parameters will describe the time course of the drug in the body. So how much time your drug will be residing in your body, all these parameters will explains you, and they do not change even with single or multiple dosing. So with irrespective of this single or multiple dosing, these parameters may explains you the how much time your drug will be staying in your body. So they may unaffected by the dose. Only those are concentrating on the time course of a drug. These are not concentrating on the dose. That's why these parameters may be unaffected by the dose. Those may be considered only the time course of the drug in the body. So when this superimposition occurs, the pharmacokinetics of the drug is said to be dose dependent or linear. Generally, our aim is this ADME is superimposed to derive the PK parameters. Okay, with irrespective of single or multiple dosing. But if you want to derive this kind of pharmacokinetic parameters, this will be dependent upon the time course of the drug in your body. So, when the superimposition process will be occurs, that pharmacokinetic of drug will be said to be dose dependent or linear. But in this case of these parameters will not get affected by the dose. See, don't get confused. Here they may unaffected by the dose I written, and here these are dose dependent. Don't get confusion. So this general mechanism of this ADME when superimposed to this PK parameters. These PK parameters will travels you in your body depending upon the time course of the drug. Okay, but these are not depending upon the dose. When this superposition is occurs in the pharmacokinetics of the drug, this will be said to be dose dependent or linear. So when you derive these kind of pharmacokinetic parameters, these only said to be unaffected by the dose. Only these may be affected by the time course of the drug. So this is of your all linear pharmacokinetics. And then what do you mean by non-linear pharmacokinetics? See, the rate processes of drugs ADME are depend upon carrier or enzymes that are substrate specific and have definite capacities and susceptible to saturation at high drug concentration. If you take more than the prescribed dosage form, what it will get? It will have some saturation, okay, at high drug concentration. In such cases, and essentially first order kinetics will transforms into a mixture of first order and zero order. Then the pharmacokinetic parameters of the drug will be changed because of the administered dose. Suppose you want to take 100 mg only, but you have taken that 15 mg. So it leads to change its pharmacokinetic parameters because of the transformation of first order kinetics to first order and zero order processes. So these pharmacokinetics are said to be dose dependent, or term synonyms are mixed order or nonlinear or capacity limited kinetics. Only one thing here you are going to remember that these ADME mechanisms or processes may have some saturation at the high drug concentration. When you take the required, instead of taking the required amount of the drug, when you take high drug concentration levels, at that point there is a chance to get saturation. So in that, it may leads to produce, instead of first order kinetics, it is using to produce first order and zero order mixture. That's why it is called mixed order kinetics. Okay. See how it will be. So why this non-linearity will be happening, and what are the tests, what are the determinations to get your non-linear pharmacokinetics? There are several tests to detect the non-linearity in the pharmacokinetics, but the simplest ones are first test and second test. First one, how you will determine this non-linearity? Determination of steady state plasma concentration at the different doses. This is one point. So you are taking 15 mg instead of taking 10 mg, right? So here 5 mg difference is there. So for the first time you need to calculate the CSS for 10 mg and then again for 5 mg CSS. So how much time it is reaching to produce its steady state concentration. Okay, so in that pattern, determination of steady state plasma concentration at different doses. This is one of the method, and the second method is determination of some important pharmacokinetic parameters such as fraction of bioavailability, capital F, elimination half-life, KET half, elimination half-life, and total systemic clearance, CLT, at different doses of the drug. So you need to calculate all these pharmacokinetic parameters for each and every dose. Any change in these parameters is indicative. It is said to be non-linearity. So you need to calculate F, KET half, CLT. These three parameters are important to tell you about whether it is following non-linear or linear. So how it is happening in case of drug absorption. Okay, let me discuss one by one how it is in absorption and how it is in distribution and metabolism and excretion. You have to read and write all these four causes of non-linearity. So in case of drug absorption, how this non-linearity will be happening? There are three causes for doing this. So what are those? Solubility or dissolution of the drug is rate limited. So for which type of drugs this phenomenon was happened? Once you recollect your drug absorption topic. So which type of drugs will exhibiting this kind of phenomenon? Solubility or dissolution. If the drug is having more soluble, the dissolution is also more. So in this phenomenon will be said rate limited. In case of poorly water soluble drugs, griseofulvin is poorly water soluble drug. So this is said to be solubility or dissolution rate limited. At high drug concentrations in the intestine, it will undergo dissolution or solubility. There is an alteration of its drug absorption. Why? We are considering it as a poorly soluble drug. But what it is doing? It is enhancing its solubility or dissolution, where at high drug concentration in the intestine. So it is exceptional. Okay, we are thinking that it is a poorly water soluble drug, basically doesn't shows any solubility or dissolution. So definitely we will say that it is following linear kinetics. But it is showing at high drug concentrations, it is showing solubility. Then definitely there is a alteration in its alteration in its linearity. That means it will produce some non-linearity. Okay, so that is one cause. And then the carrier mediated transport system. When this carrier mediated transport system will occur in case of active transport of the drugs. Okay, all these things we studied under absorption. So when you want the process of active transport mechanism for actually transporting your drug, okay, into the biological membrane, you have a need of carrier. So this carrier mediated transport system, few drugs like ascorbic acid is saturated with the carrier, with the particular carrier what you are going to use for its transport system. So in that cases, there is a saturation of transport system will occurs. This is what the deviation from the linearity. Then it is showing the non-linearity. Okay, so most of the drugs are inactive to transport through biological membrane. In that situation, there is a requirement of a carrier to get its active transportation through the cell membrane. So in that cases, few of ascorbic acid like drugs will get saturated with the type of carrier. What the type of carrier we used? So if it will get some saturation, then there is a alteration in the linearity, then it is showing its non-linearity. And one more important thing, pre-systemic gut wall metabolism or hepatic metabolism. This is in case of metabolism. See, pre-systemic gut wall or hepatic metabolism, it will attain some saturation in case of lol drugs, propranolol. So these are a few exceptional causes, okay, few exceptional causes which will exhibiting this non-linearity. And the pharmacokinetic parameters will get affected because of this non-linearity are F, Ka, Cmax, AUC. All these four pharmacokinetic parameters will get affected because of the non-linearity have seen in this case of drug absorption. And then a decrease in these parameters is observed. In the former two cases, there is a decrease in the parameters. And then this in case of pre-systemic gut wall or hepatic metabolism, it will be increases. That is, the drug absorption is more than that of expected absorption. So these two, based on these two parameters, we will say that these drugs will follow some non-linearity based on the deviations what we observed under drug absorption. And the other cause for this non-linearity is changes in the gastric emptying and also the gas GI blood flow. So these are other changes and you have to focus only on these three major changes. Because of these three changes, the drug will have nonlinear kinetics in its absorption. And then in case of distribution, why the nonlinearity was happening. See, this is also in case of two cases. First one is the binding sites on plasma proteins get saturated. So what the drug is go and bind to one plasma protein binding ligand, it will get saturated with that ligand. Then there is an alteration in its drug distribution. If the drug is binding to its ligand, what is happening here? It is increasing its half-life. Why? With this of ligand, the drug will be bind, then it will take some time to distribute itself in the circulatory system. So definitely it will increases the half-life of the drug. So if the drug will get saturated with this ligand, then there is a deviation of your drug distribution. There is no chance to get increased half-life and definitely it will show some non-linearity. The example for this case is phenylbutazone. Examples of the drugs are very important. Phenylbutazone. It is showing that binding site of phenylbutazone on plasma proteins will get saturated. Because of that, there is a deviation from its distribution pathway. And then what are the tissue binding sites get saturated in case of naproxen? In case of naproxen, the tissue binding sites available in this naproxen will get saturated. So these two are the binding sites of plasma proteins and tissue binding sites will get saturated. That's why these will show some non-linearity. And in both cases, there is increase in the plasma drug concentration. Yes. So these may get saturated. These are not focusing on their distribution. Only these will focus on the saturation point. So once the drug will get saturated with the binding sites or tissue binding or plasma protein, what is going to happen here? It is increasing its concentration. It is increasing its concentration. So that's why there is an increase in VD we have observed in binding sites and plasma proteins, and the clearance with high ER. ER means extraction ratio, what we studied under clearance. Clearance with high ER get increased due to the saturation of binding sites. If the plasma drug concentration is increased, then there is a chance to increase its clearance also because of the saturation of binding sites. And only the unbound drugs, only the unbound, that is free from binding. Okay, this unbound clearance of drugs which are exhibiting low extraction ratio, these may be unaffected by this nonlinearity and they will show its pharmacological response. Here you need to remember only the binding sites on plasma proteins and the tissue binding sites. Because of the saturation we occurred in these binding sites, there is a chance to increase its plasma drug concentration. Then in the clearance with high ER get increased due to the saturation. So the free bound or unbound drugs, clearance of drugs with low ER will not be affected by this non-linearity. Okay, this is of drug distribution. So how about metabolism? In case of metabolism, non-linearity will occur due to capacity limited metabolism. Small changes in dose administration. There is a large variations in the plasma drug concentrations at steady state. So you might have seen this inter-subject variability. So what it is saying, here there are two important causes. Here you need to focus only on capacity limited metabolism. The enzyme or a cofactor saturation will be seen. See, phenytoin came here. Phenytoin, sodium and alcohol. These will exhibiting capacity limited metabolism. Okay, so instead of taking small doses, if you take the large dose than the prescribed dose, okay, what is going to happen? You may have seen a large variations in the plasma concentrations at the steady state. So it will shows you the large inter-subject variability. That means subject to subject, inter-subject. Okay. So if you want to do some animal studies or human studies for this phenytoin sodium, so if you take 1, 2, 3, 4, 5, 6 numbers of animals or human beings, so you may observe the inter-subject. Suppose it is showing 10 microgram per ml concentration and it is showing 8 microgram and it is showing 12 microgram. So this is of there is a large inter-subject variability was observed because of the capacity limited metabolism of this phenytoin. Okay, so this is called as inter-subject variability. Subject to subject, we are going to find out some concentration variability. Okay, this is called the inter-subject variability. So the two important causes for this drug metabolism. First one is capacity limited metabolism. Either it may be through this enzyme or cofactor saturation. Examples are phenytoin and alcohol. And one more important thing is enzyme induction. So in case of enzyme induction, there may be a decrease in the plasma concentration. The example for this is carbamazepine. So apart from these two important causes of drug metabolism, you may have hepatotoxicity and the decreased hepatic blood flow. So, in case of hepatotoxicity, acetaminophen drug will produces these changes in the metabolism. Why? Because this is producing hepatotoxicity. And in case of decreased hepatic blood flow, we observed this hepatic blood flow decreased in case of propranolol. So, there is an alteration of its drug metabolism. So, so in these cases, the drug will deviating from its linearity. So we want this, but it is not going like this. It is deviating. It is showing some non-linearity. Okay, because of these parameters. Induction in dose dependent concentration. See, saturation of the enzymes will decreases you the hepatic clearance and increase in the steady state concentration. So if it will be increased beyond the level, it is also one of the toxicity symptom. And in case of enzyme induction, it is a reverse condition. We studied about enzyme induction and enzyme inhibition phenomena in the metabolism, right? So the other reaction, other reasons which are includes to deviating from its linearity, saturation of binding site, inhibitory effects on the metabolites, on the action of enzymes. Okay, this is of metabolism causes non-linearity, and only the last one is drug excretion causes non-linearity. Why this drug excretion will causes non-linearity in the pharmacokinetics? There are two processes which are saturable. These two processes will get saturable, then it will be deviating from its linearity. What is that? Active tubular secretion. The famous example is penicillin G. Active tubular reabsorption, glucose and water soluble vitamins. Saturation of the carrier systems. What is going to happen once the carrier system will get saturated? There is a decrease in the renal clearance in case of this active tubular secretion. So in case of this active tubular secretion, the name is active means definitely it is using some carrier for its mechanism. So this active tubular secretion, there is a saturation was happening in the carrier system. Then decrease in the renal clearance was observed. And in case of number 2, that is active tubular reabsorption, what you have seen, drugs will undergo saturation in the which is used in the treatment of asthma, and in case of nephrotoxicity, aminoglycosides, in case of saturation of binding sites, disopyramide, and in case of biliary excretion, non-linearity will be due to the saturation. This may be seen in case of tetracyclines and indomethacin. So these are of different kinds of drugs which will exhibiting the nonlinear pharmacokinetics because of the excretion processes, because of the unwanted excretion processes like forced diuresis. In case some drug will be excreted unwantedly, and if the urine flow will be increases, definitely there is an excretion. Then nephrotoxicity, and in case of saturation with binding site. Only one thing you have to remember here: if the drug will follows exact ADME as per the description of in the PK kinetics, it is showing linear kinetics. If it is having any deviations from either of absorption, distribution or metabolism or excretion, if it is showing any deviation in the sense how it is showing, in case of absorption, we need more solubilized drug, we need more dissolution, then we will get some expected absorption. So if the poorly soluble drug will get more solubilized in our body, then definitely there is a deviation in the absorption we have seen. So in that cases, in case of drug distribution, how the drug distribution will be happening, either it may be bind to the protein or ligands or plasma proteins, whatever it is. I told you already 4 to 6 plasma protein binding sites are there. So because of the binding sites, the drug will exhibiting its pharmacokinetics very linearly. Either there may be having any deviations in case of naproxone like that drugs. So it is showing some non-linearity. So in case of metabolism, it is not get metabolized clearly. Okay. So then it is showing some deviations. In case of excretion, we have seen some unwanted excretions in these types of drugs. So these all will says you that deviations or saturation of the drug at high drug concentrations in the body, the drug will get saturated. So that's why it is not able to produce the linear kinetics. It is able to produce only the non-linear kinetics. So these four are the major causes of the non-linearity of this ADME. Okay, because of this non-linearity with this ADME, we have observed the non-linear kinetics or mixed order kinetics or capacity limited kinetics. Okay, so next class I will tell you all these things. These are of some examples of the drugs which will shows the non-linearity clearly. See here, these are the causes. Either they may have GI absorption or distribution or metabolism or excretion. This is excretion, not elimination. Okay. And then these are the drugs. You just learn this table. Saturable transport in the gut wall. The example is riboflavin, gabapentin. Saturable GI decomposition, penicillin G, omeprazole. So in this pattern, you have to learn all these examples of the mechanisms or causes of nonlinearity and the drugs for these examples, which type of mechanism it is following. So it is very simple and very easy. Once you read it, then it is easy. Okay. So these are the few examples of the drugs which are exhibiting the nonlinear pharmacokinetics. Okay.